Okay. Okay, so good evening and good morning, everyone. So we today we have a treat. It's really my great pleasure to welcome today's speaker, Dr. Rafi Ahmed from Emory. Rafi is currently a Charles Howard Chandler Professor in Emory University. So I think most of us probably know Rafi already, but it's just a very brief introduction. So Rafi completed his undergrad student study in India in chemistry, and also part of it study was complete in Idaho State University in US in microbiology. And then afterwards, Rafi completed his PhD training in microbiology and molecular genetics in Harvard University. So Rafi actually started his postdoc training in Scripps in Department of Immunology, and then start his own independent research program in Department of Microbiology and in Immunology in UCLA, and all the way up to full professor in UCLA. So from 1995, Rafi actually moved to Emory University School of Medicine in Department of Microbiology and Immunology as a full professor, and then also as a director of Emory Vaccine Center School of Medicine. As I just mentioned, that from 2010, Rafi is Charles Howard Chandler Professor in Emory University. So I think, as I said, Rafi has so much great contribution in science. He's a member of National Academy of Science, also a world-renowned immunologist whose work during the past decades has been highly influential in shaping our current understanding of memory T cells, differentiation, and antiviral T and B immunity. So the general goal of Rafi's lab is to understand the machinery of T and B cells, immunological memory, and to use this information to develop new vaccines for the prevention and the treatment of diseases. Rafi's lab studied antigen-specific immunological memory in both murine, in primate, and the human system. So another focus in Rafi's lab is to develop strategies to restore function in virus-specific T cell during chronic virus infection, such as HIV. And in particular, the groundbreaking discovery of inhibitory receptor PD-1 as a major mediator of T cell dysregulation during chronic infection. I think a lot, probably everyone have read Rafi's work. Another contribution from his lab is to understand the humoral memory development and maintenance, and to develop new methods for rapid generation of human monoclonal antibody after vaccination, and have been shown that broadly cross-reactive antibody that recognize multiple influenza virus can generate after influenza vaccination in humans. So as I said, Rafi's actually have a lot of groundbreaking discovery contribute to scientific community. And based on these really distinguished contributions, Rafi have been awarded by numerous of honors and awards. I'm not going to read all of them. It's just an entire page. I think everybody knows that Rafi is a member of American Academy of Art and Sciences, also a member of Academy of Sciences, and then also a National Academy of Medicine. 2017, he awarded as a William Collier Award in Cancer Research Institution. He's also a member of National Academy of Inventors, Distinguished Fellow of American Association of Immunologists. Besides these awards, I think the most impressive part is Rafi have over almost close to 500 different publications. But last but not least, he's really a great mentor who coach and guide a lot of outstanding trainees who are currently active in both academia and the industry. I personally was super impressed by not only his dedication in science, but also his humbleness and mentorship for the young generation of scientists myself included, of course. So I think without further ado, we are very much looking forward to your talk, Rafi. Thank you so much for doing this for us. Please go ahead.
Thank you. Thank you, Chad, for the very kind words. So what I'm going to do today is describe to you some of our recent work looking at how CD8 T cells adapt to a chronic viral infection or a chronic antigen stimulation. So my talk is in three parts. In the first part, I'll give a kind of an overview of published work by us and also by many other outstanding groups, which have defined really the different CD8 T cell states during chronic viral infection. So most of the data that I'll summarize in this first part will come from our studies using the mouse model of chronic SCMV infection. So the basic principles of T cell differentiation have been defined using this model. I will then switch to kind of an overview of work we've recently done on CD8 T cells in human cancer and give you a bit of a summary of what we've done in lung cancer patients. And then spend a little bit more time describing our recent work looking at HPV-specific CD8 T cells in head and neck cancer in patients who are HPV positive. I will then end on the last section, which is implications of understanding of the CD8 T cell differentiation pathway for developing immunotherapy. And I'll describe to you some of our recent work where we have used PD-1 plus IL-2 to actually change the differentiation trajectory that one normally observes during chronic infection. So this slide is a kind of a busy summary slide, but it kind of puts together in a cartoon fashion the current understanding of CD8 T cell differentiation during chronic viral infection. The work from our lab actually was done here by Seijin In, who was a postdoc in the lab, and he's now on the faculty at a university in Korea. And at the same time, also Lilin Li from China, also lucky to have Lilin as a former postdoc in the lab, but independent work by Lilin also made similar findings. And actually, Seijin's and Lilin's papers came out together in this 2016 issue of Nature. So the key finding that came out of our lab, Lilin's lab, and also several other labs that followed up on our work is that there is a very unique population of CD8 T cells that one finds in the context of chronic infection. These cells arise fairly early. They arise within a week after infection, coming from naive CD8 T cells, but then there's a nice bifurcation that gives you more effector-like cells. But then you generate this very unique population. I'll refer to this as stem-like CD8 T cells, or also use the term chronic resource cells to describe them. The term precursors of exhausted cells or progenitors of exhausted cells is also used by many people. And when we say PX, or I'm saying resource, chronic resource, or stem-like CD8, we're talking about the same cell. So what are the unique characteristics of these cells? Probably the most interesting character of these cells is their location. So these cells are predominantly found in lymphoid tissue. Like, for example, in this mouse model of chronic SCMV infection, the virus is everywhere. It's in pretty much all of the non-lymphoid tissues, including some of the lymphoid tissues, but it's everywhere. But these particular type of cell is only found within the lymphoid system. That is, they'll be in the spleen, in the lymph nodes, a few in the bone marrow, but the major site of these is the spleen and the lymph node. And within the spleen and the lymph node, they're found in the T cell zone, not so much where there is active high-level infection, but within niches in the T cell zone. And I'll be coming back to this theme of these cells being in niches. So these cells are very quiescent. They undergo a slow self-renewal, but they're quiescent. But they're very pluripotent, have proliferative potential and differentiation potential, but they're very quiescent. As I said, they are in mostly lymphoid tissues. Also, they're resident there. So they do not circulate into the blood. When there is a heavy chronic infection of these mice, you will not find these cells in the blood. They're resident in the lymphoid tissue. They express PD-1, and we know that they are getting TCR signals, but they still don't differentiate. They maintain this quiescent state. They express TCF1, and TCF1 is essential for their generation. If you have cells that don't express TCF1, then basically you will not generate the population. But they also express BCL6. So they have kind of a little bit some aspects of the T follicular cell phenotype too. But of course, their properties are very different. They express high levels of co-stimulatory molecules. The other inhibitory receptors are lower on these. They have PD-1, but the other ones are lower. They express interesting, and they have no effector molecules. This is the key thing. There are no effector molecules in these cells. They express a very interesting sets of chemokine receptors and secrete interesting chemokines. And I just point out a few of them. So they express CXCL5, so they can migrate in response to CXCL13. They express sufficient levels of CCR7. They're actually quite low overall when you compare to naive cells, but there's enough CCR7 that they will respond to CCL19 and CCL21. Okay, again, these are things which keep them in the T cell zones. They express XCL1, kind of a unique thing that other CD8 T cell subsets don't do. They express XCL1. XCL1 will attract the XCR1 DCs, which are your cross-presenting CDC1. And if you look histologically, they will be surrounded by these XCR1 DCs. So these cells are continuously at a slow rate. They're continuously generating what I will refer to as transitory effector cells. So when this differentiation happens from the stem light to the transitory effector, the first thing that happens is downregulation of TCF1. TIM3 comes up, TBET comes up, basically the effector programs comes up, and they come out in the circulation. So for example, these will be CD69 high, these will be CD69 low. And S1PR1 will be expressed by these cells. There's no S1PR1. So these are resident cells. These cells, the transitory effectors emerging from these express effector molecules, start proliferating, come out into the blood, will go to sites of infection, and reasonably have reasonably good function, even though PD-1 is still inhibiting their function. Eventually, these are the cells that become kind of terminally differentiated or exhausted. Again, these cells also become resident. This is the one that one finds in the blood. And then they will go to the tissue. So these more exhausted cells that we and many others have defined have very low levels of co-stimulatory molecules, multiple inhibitory receptors, but they still have effector molecules, but not very functional because of the many inhibitory molecules kind of keeping them in check. These transitory effector molecules were defined by Will Hudson, the postdoc in the lab in Immunity in 2019, and Wegu also independently showed them. So the Wegu's paper and this paper. This migration differences were shown by Sage in a PNS paper. So what happens when you do PD-1 blockade? So this slow differentiation is happening continuously. When you do PD-1 blockade, you now get much more increased differentiation of these cells. So you can get a 5-fold or up to 10-fold increase in the generation of these transitory effector cells. Many more of these cells are generated because the PD-1 break is removed. They come out into the blood and will go to sites of infection. And then you have PD-1 acting at the site of infection. So you block the PD-1 PDL1 interaction at the target site. And that results in efficient killing of the target cell and more cytokine release. So I think everyone appreciates now quite nicely that the proliferative burst after PD-1 comes from these cells. That's why I like the term resource cell, because this is the one that's generating continuously at a slow rate, remove the inhibitory break, they generate many more of the transitory effectors. What is less appreciated is that PD-1 blockade is also important at the target site, i.e. PD-1 has to work on these cells and also maybe some of these cells so that they can elaborate effector function and kill the target cell. So PD-1 is acting at two levels. One, acting on these cells to provide the proliferative burst and acting on these cells for actually target cell killing at the tumor site or at the site of the viral infection. Okay, the next slide is just a few genes that are expressed in the three different subsets. All of this is published by us and many people, but I thought it would be good to show you one single cell RNA-seq example of how these three subsets look, how they differ from each other. So this is single cell RNA-seq of tetramer sorted cells. So this is, we go into a chronically infected mouse, use the appropriate tetramer, sort the tetramers of positive cells from the spleen, and then do single cell analysis. And naive cells are spiked in here as a comparator. So the spleen is a very good site to look at all three subsets, because within the spleen, in the T cell zones, you have the stem-like cells. The most of the infection in the spleen is in the red pulp, in the F480 macrophages. So in these sites of infection, which is the red pulp of the spleen, you will find the more transitory and exhausted cells. So in the spleen, you find all three subsets. It's a very nice place to do single cell RNA-seq and describe these three subsets. So here are the three subsets. This is the stem-like, the transitory, and the exhausted. All of them express PD-1. All of these also express STOX. Work done by John Berry and many others. Andres has shown that the STOX is an important part of the exhaustion program, and you see that all of them. So based on TOX or PD-1, there is not much distinction of these three cell types. PD-1 is slightly lower in stem, but again, very high compared to naive cells and memory cells. All express PD-1, all express STOX. But now look at this, TCF1 only expressed by this cell population. So there's no TCF1 here or here, because TCF1 gets downregulated in part of this exhaustion program when these cells differentiate. Naive cells have TCF1. I should really emphasize this. TCF1 is found in all naive cells have TCF1. Pretty much all memory cells after acute infection of TCF1. So TCF1 by itself is not a marker of these cells. You would have to combine this with these cells also having TOX, and most importantly, these cells also being PD-1 high. So if you're looking for cells of this nature, they would be PD-1 high and also TCF1 positive. The PD-1 part is very interesting because PD-1 comes up on TCR stimulation. So what this tells you is that there is an antigen of interest that these cells are seeing, and that's why they're PD-1 high. So again, in the context of tumor in humans, and also in the context of chronic infection, these cells will be PD-1 positive and also TCF1 positive. And these would be the canonical cells that we would call the stem of the resource cell. Again, TIM3 is on these two populations, the transitory effectors and exhausted, but not on the stem-like. 39, another marker of exhaustion, but not on the stem-like cells. Porphyrin effector molecules in these two cells, but not here. Again, granzyme B not here, but in these two populations. In contrast, CD28, an important co-stimulatory molecule, is expressed more on the stem-like cells and decreases as you go in this direction. And CD127, which is critical for survival of the cells, is expressed only by the stem-like cells. It's lower level than naive cells, but it is enough there to keep cells alive. So again, a key thing about T-cell biology is that if a cell does not express CD127, it's going to be a short-lived cell. It may live for a few days or for a few weeks, but then it's going to die because the IL-7 signals are critical. And within these three cell populations, only this one is the long-lived cell. Both of these are short-lived cells. That's another reason why there has to be continuous differentiation to keep the response ongoing. That's why I personally prefer the term chronic resource cell, because they're feeding this pathway, because the transitory effectors and exhausted have a finite short lifespan, whereas the stem-like cells can self-renew at a slow rate and have a much longer lifespan. So keep feeding the system. And again, XCL1 only expressed by the stem-like cells, not by these, not by the naive cells. Okay, so with that background, I just want to, before I get into our human tumor work, I just want to acknowledge many of the other papers that were published around the time of our paper came out. I mentioned to you Lillian's paper, and then there was also a very nice paper from Dietmar Zane, which came out in Immunity around the same time, independently describing the same population. And then after our papers came out, there were a series of papers describing it in other viral systems and extending some of these kind of observations. And then very quickly, and I'm just citing here the early papers that came out. Now probably several hundred papers have come out, kind of describing in cancer, human cancer, things that look very similar in terms of the differentiation. All right, so let me now spend a few minutes very briefly summarizing some work that we've done in lung cancer, non-small cell lung cancer patients. The question basically here was, do you find stem-like CD8 T cells in human cancer? And the answer is yes, we do find them in both. And I'll just show you a couple of examples without too much data over here, and then a little bit more time I'll spend on the HPV positive system. So there were two, a former postdoc in the lab, Alice Camphorst, who's now on the faculty at did this work. And an interesting finding that Alice made, which was very consistent with our understanding of the CD8 T cell states from the mouse model studies, is that after PD-1 therapy of lung cancer patients, this was actually among the first papers to show this, that there is a burst of effector-like cells that come out into the blood. In this case, we do not know the antigen specificity, and at that time, we did not characterize any new antigens for the CD8 T cells, but we looked at the pre and post. We looked at the percentage of CD8 T cells which were effector-like, i.e. they had granzyme B, they had perforin, they were proliferating, expressed CD38, HLA-DR, all canonical markers of activated effector-like CD8 T cells. So at the baseline was quite low, but within 2 weeks of the first infusion of PD-1, there was a significant increase in the number of proliferating CD8 T cells. And this increase in proliferating CD8 T cells correlated with the clinical outcome. So what we showed in this study is that about 60% of the people respond with a burst of CD8 T cells that come out. And out of these 60%, about half of them show a clinical response, whereas the ones that do not show this proliferative burst, hardly any of them show the proliferative response. So this paper established two things. Effector-like CD8 T cells come out in the blood after PD-1 blockade of lung cancer patients. And second, that the magnitude of this can be an indication or a biomarker for the outcome, clinical outcome of these individuals. Alice then followed up with a very important observation, which was, this paper was published in Science in 2017. And this was a combination of mouse experiments and also human lung cancer studies. And this paper showed that removing the PD-1 brake is essential for getting this proliferative burst of cells coming from the resource cells, but it was not sufficient. So in addition to removing the PD-1 brake, the cell also had to get co-stimulatory signals through CD28. And if only the PD-1 brake was removed and the CD28 B7-1 interaction was blocked, then there was minimal to no differentiation of this. And this is very consistent with two things. One, the expression pattern of CD28, because again, the stem-like cells have CD28. And I emphasize this, the niche that they're in. So they are surrounded with DCs. They are sitting in a sweet spot within the T cell zone where DCs are there. And so they get then B7-1, B7-2 signals through CD28. And these cells also have CD28. So this showed that co-stimulation and CD28 expression was also important in the biology of these cells. I'll show you very quickly some gene expression data, which many others have also shown. So this is now isolating straight from the TILs. This is a lung cancer patient who's having surgery. This is not a lung cancer patient having PD-1, just having surgery. So from the TILs, you can isolate PD-1 positive cells, separate them based on CD28 or other markers into the more exhausted-like cells and more stem-like cells. Then if you do RNA-seq on them, you see that these more TIM3 positive, CD39 positive cells have many more inhibitory receptors. They don't have CD28, but the stem-like cells do have CD28. These cells have effector molecules, these don't. But most importantly, now if you look at TCF1, these cells have TCF1, these don't. I should also point out that LEF1, which is another important transcription factor, is found in these cells and not in the more terminally differentiated cells. Okay. And of course, BCL6 is here and not here. Okay. And again, CD127 by these cells and not by that. So pretty much the major observations that were made by us and others in the mouse systems kind of started correlating with the subsets that one finds in lung cancer. Okay. But the next question I want to address was probably the most interesting one. I mean, I've told you until now that these cells, at least in the mouse system that we study, that these cells are found in lymphoid tissue, the i.e. spleen and lymph nodes. But here, I'm telling you that if I grind up the lung tumor, basically we were getting a piece of the lung tumor, we were not getting any lymph nodes in the study, no draining lymph nodes were analyzed in the study. Almost certainly some of those cells will also be there. But in this case, we're just grinding up the tumor, and I'm telling you that there are stem-like cells in the tumor. So the question is, where are these stem-like cells in the tumor? What is their anatomic location? And I think that these data are really quite nice, that I'll show you in the next few slides. So what is the location of the PD-1 positive, TCF1 positive stem-like cells in the tumor? Now I'll tell you the answer and then show you the data. Basically, these cells are found in tertiary lymphoid structures. So tertiary lymphoid structures, they can either be within the tumor itself, or they can be just outside the tumor, or even a little bit further away. But if I show you a couple of slides looking at intratumoral TLS, then you'll see that this population is localized in the TLS region, not in the, you won't find these cells on the tumor stroma, but they will be again in this niche within the tertiary lymphoid structures. So here is looking now, in this case, we are looking at the tumor itself. So this is, here is the tumor based on cytokeratin staining. This is tumor, all of these. This tumor area has a lot of CD8 T cells in there, and pretty much all of them express PD-1. This means that they're seeing some antigen from the tumor. So a lot of CD8 T cells, a lot of PD-1 positive cells on this tumor, hardly any TCF1 positive. And these cells really have also other effector molecules there. Then in the next slide, I'll show you where there is a tertiary lymphoid structure within the tumor region. So here's now the tumor, and this, what is appearing as a black hole here, is actually where the tertiary lymphoid structure is. Based on H&E, you will find B cells, CD4 cells, CD8 cells, and also dendritic cells in this area. So now you ask, where are the CD8 T cells? They're everywhere. Then you ask about PD-1, all of these cells express PD-1. Then you ask which of these CD8 positive, PD-1 positive express TCF1, and you see that these are right sitting in this hole over here. So the stem-like cell, the TCF1 positive, PD-1 positive CD8 T cell, is not on the tumor bed. It is within the tertiary lymphoid structures, which are right in the tumor region. It's when these cells differentiate, they will become more effector-like. Then the program will be to go to where the tumor is. And that's exactly what is happening in the LCMV mouse model. We first described it. The white pulp area is not heavily infected. Probably some antigen there that's kind of keeping them through some cross-presentation mode. Okay. But it's only after they differentiate, they'll go to where the virally infected cells are. Quite amazingly, we find this beautiful identification of these cells in the tertiary lymphoid structures. And there's now data coming out from several studies showing that the TLS may correlate also with responsiveness. This is soft data that's coming out from different studies because there isn't such fantastic quantification of TLS in the tumors. But the data is emerging that the presence of TLS has a good, may have a good indication or good correlation with PD-1 blockade. In other words, what that means is that you're likely to have more of these stem-like cells in that individual. Okay, I want to now switch to an effort that we really wanted to put in, which is defining antigen-specific T cells. So far, what I've shown you is really just total CD8 T cells. And actually, most of the very nice cancer papers which have come out have also looked actually at total PD-1 positive CD8 T cells. There have been a few nice papers looking at neoantigen-specific cells, but in general, the analysis has been on total TILs and not necessarily using MHC class 1 tetramers. So we wanted to really do a deeper dive and look at antigen-specific CD8 T cells in human cancer. So what we did here is that we picked HPV positive head and neck cancer as our system to study this, because then we knew which the viral antigens are, much easier than defining new antigens in individual people, and because then each person is different. So here with the viral mediated cancer, we could actually look for the same epitopes, and as long as the HLA matches, we could look in several people. And this project has gone really well. Andreas Wieland, who was a postdoc in the lab and is now on the faculty at Case at Ohio State, played a major role in it. Another postdoc, Christian Eberhard, who's now back in Geneva as a faculty member at the University of Geneva. So they basically led these studies and looked at CD8 T cell responses in human head and neck cancer patients. And so this was our approach. Our approach was the following. This just shows you that HPV is a major cause of oropharyngeal cancer. And actually now in the US, about 50 to 70% of the oropharyngeal cancer actually are due to HPV. And HPV-16 is the major HPV type that does it. So our approach was the following. Since we did not know which responses would be in these individuals, we first had to define which responses these individuals might have. So what we got was, we got sample from treatment naive patients. This is the key point too, and I'll come back to this later. So these were patients who were diagnosed with head and neck cancer. They were HPV positive and were going in for surgery. They've not had any treatment. They've not had chemo. They haven't had PD-1 or any of these treatments. They're going in for surgery. And we were able to get we were able to get the primary tumor from these individuals. We collected blood from these individuals. And then we were also able to get a metastatic lymph node from these individuals. So we basically had from each of these patients 3 samples. We had the primary tumor, the metastatic lymph node, and the blood. I want to emphasize that this is not a draining lymph node. This is a metastatic lymph node. So basically the tumor sample we got was almost pretty much all tumor. So we could basically looking at 2 different tumor sites and in the blood. So what we decided after we got these samples, our strategy was that from the blood, we try and first identify which HPV proteins these individuals might be seeing. So what we did was we took the blood of these individuals and then expanded these cells for 2 weeks with 251 predicted HPV peptides coming from E2, E5, E6, and E7. These are all HPV proteins. E6 and E7 are the oncogenic proteins. So every cancer cell that's transformed by HPV will express E6 and E7. But we also include, and the major focus of the field is on E6 and E7 as target antigens, but we also included E2 and E5. And because there was some data in the literature suggesting that some of the HPV positive cancer patients may also have E2 and E5 specific T cell response. So basically, we use this pool of peptides from these 4 proteins and then stimulated the blood from these cancer patients for 2 weeks and then checked for the presence of HPV specific T cells initially by interferon gamma ELISpot, then by additional cytokine staining. Then we defined the exact peptide which it is, looked at the HLA binding, did the HLA mapping of these individuals from the blood, and then did tetramer staining. I should first tell you that in all 17 patients, we saw a response. So there wasn't a single patient that was negative when we did the interferon gamma ELISpot, i.e., they all had some T cell responses to some combination of these. But we did the deconvolution in the ones where the responses were slightly higher. So here's an example of a gamma interferon gamma ELISpot after the expansion. This is then showing you by intracellular cytokine staining that these are indeed CD8 T cells that are making this. And then identifying the peptide, knowing the HLA, and then showing with the tetramer that you have a beautiful staining with the tetramer. So we knew in the subset of these patients which were the cells which these patients had in them. And we ended up identifying several epitopes, and the ones in the bold are the ones that I'll show you some data from. Then we went to the real question. And the real question was, if we now look in the tumor, so this is all blood, we have expanded from the blood, and now the question was, if we look now directly in TILs, will we find, what will we find? Will we find any of these antigen-specific cells in the tumor? And of course, this is something that we had not done before. In fact, no one had done it until now for HPV and looking in the tumor itself. Okay. And we were very pleasantly surprised. We found that actually not only were these cells present, but they actually are present at fairly high frequency. So here's an example. Now this is direct ex vivo, no expansion. So we have the tumor, we had the primary tumor, the metastatic tumor. So we grind up the tumor and we look at, use the tetramer and look at the response. You see that in this patient, there is a AO1 restricted peptide presented from the E2 protein. And if you look in this individual, 3% of the CD8 T cells in the tumor are responding, are reactive to single tetramer. In the metastatic primary tumor, if you look at the metastatic lymph node from that, also there at 1%. Okay, I'll come to the blood in a second. Here's another epitope, also from the E2 protein in a different patient. In this case, 5 to 10% of the, I would have never predicted that a single epitope or a single tetramer in the TILs is comprising up to 5 to 10% or 1 to 3% of the total CD8 T cells in the TIL itself without any expansion. And this is an example of some of these ranging from 0.1% to up to 10%, similar frequencies in the primary tumor and in the metastatic lymph node, as you can see, it kind of joined in the blood below our limit of detection. So this is very important because it's not that there are zero cells there. Remember, that's where we expanded it from. But in the blood, there'll be very few cells coming out because they will either be resident in the tumor or resident in some lymph nodes. Very few would be coming out in the blood. There are some there. If we had done tetramer enrichment and we had 200 ml of blood, I'm sure we would have found some, but we only had a small amount of blood, and of course the tumor sample is very limited. Okay. So, but the point here is very high frequencies of these antigen-specific CD8 T cells in the tumor as we at the time of surgery. We then asked, what does the single cell RNA-seq look like of these? In other words, do we find in this HPV human tumor TILs, will we find the 3 canonical subsets that we defined in the mouse system? And the answer is yes. You again find these 3. We did this on 13 different tetramers, single cell RNA-seq, and this is shown as a composite. It's not as beautiful as their CFP data, but you will see that you have the canonical 3 subsets there. So this is a composite single cell RNA-seq of 13 different tetramers, and you find cells that have the canonical genes of the stem-like cells. You can see that over here, TCF7, XCL1, but no granzyme B. And then these are the more terminally differentiated cells, which have the exhaustion kind of markers. And then these are the more transitory cells, which have more the effector-like and transitory markers. Okay. So basically, the 3 canonical subsets, and there probably are more subsets within these, but the 3 canonical states, i.e., the stem-like cell, the TCF1, XCL1, minimal granzyme B, but having CD127 is there in these cells, in the tumor itself. So we then ask the question, what about the functionality of these cells? So we are now identifying a stem-like population with ECF1, XCL1, no granzyme B in this. And are these cells functional? I.e., will they respond to peptide? So we then did a rather, or I should say, the people in the lab did a kind of a heroic experiment, which is sorting from the tumor two broad subsets and then looking at their functionality. So here is the experimental design for that. So we sorted from the TILs. Now we know in this case what the tetramer is, so we will know which peptide to stimulate with, and we know which tetramer to stay looking at it. So these cells were, the TILs were labeled with cell trace violet, so we can track the division of these cells. And then they were sorted into two groups very broadly into what would be the stem-like cells. These are TIM3 negative and CD39 negative. Not the most precise sorting for this, but at least it would separate it from the more differentiated cells. So that's over here. And then the more differentiated, TIM3 positive, 39 positive. Also, everything is PD1 positive. So it was basically started with PD1 positive CD8 T cells into the more differentiated population and into the less differentiated population. And then we stimulated these cells in culture for 5 days. The one culture had the cytokines, the other culture had the cytokines plus the antigen-specific peptide. Okay. And the data again were quite striking. So if you now look at the sorted stem, so on this axis is tetramer, on this axis is CTV. So of course, without any peptide stimulation, here is the tetramer positive cell, and they do not divide. Okay. But if you now add the peptide, you see this beautiful division of when you start with the, when we were stimulating the sorted stem-like cells, but with the more differential minimal to no. Essentially, these cells have the ability, coming straight from the tumor, they have the ability to proliferate and differentiate after stimulation with peptide. Here is a summary of 6 or 7 samples that were done, and you can see that in every case, the stem-like population proliferated, the terminal differentiation did not. Okay, so next question was, what about the markers? How do the markers change when these cells differentiate? And that's shown over here. So let's look at this, the stem-like population. If you now stimulate it, so unstimulated, these were all TIM3 negative, as expected. But after they proliferate, TIM3 comes up, and the granzyme B data is beautiful. They don't have granzyme B to begin with. After they stimulate and divide, granzyme B comes up because they are now recapitulating their differentiation program. 39 comes up, and IL-7 receptor, which was present on these cells, now goes down because again, this is part of the differentiation program. Effector molecule come up and 127 goes down. And if you now look at the differentiated population, there's minimal proliferation, not much happens here. These cells were already TIM3 positive, they remain TIM3 positive. They were already had granzyme B, they don't divide, they don't proliferate, it stays that way. And 39 was high, it stays that way. IL-7 receptor was not there, and of course, this stayed. So what we're showing is that these stem-like cells that we're getting straight from the tumor, antigen-specific cells, when you provide the peptide, they have the ability to differentiate and become more effector-like cells. And here are some additional markers just to point out that everything is RO. They're all RO positive. CD25 comes up, which again is this very important post-stimulatory, activation marker. And these cells doesn't. CD28, an important one. The stem-like cell had CD28. The terminally differentiated exhausted cells did not. And when they divide, many of them still retain it because this is the early emerging effectors from this will still retain CD28. And then the last data slide here. So where are these cells within the tumor? And again, the data is very nice. The stem-like cells are again staying away from the tumor. So here is a tumor histology. This is all tumor. This is staining with cytokeratin. And in this case, we just kind of mark this area to tell you where the tertiary lymphoid-like structure was. So here are CD8 T cells. They are on the tumor as well as in this area. And PD1 is also everywhere. And the TCF1 beautifully located in that area. Again, so they are here and not in the tumor. So this is the beautiful biology of these stem-like cells. That's how they conserve that's how they preserve their quiescence and the ability to remain in this niche. And only after they differentiate will they go to where the tumor is expressing effector molecule. So I've shown this to you starting with the mouse model experiment that we did and published, that they are in the T cell zones, and then in the lung cancer patients, in the tertiary lymphoid structures. And again, over here, these cells are in what our pathologists prefer to call the tumor stroma and not in the tumor parenchyma. That is, this is the tumor parenchyma, this is the tumor stroma. All right, so let me summarize this. Both of these papers, this paper was published in 2021. Christian Eberhard actually was the first author of the CD8 T cell paper. Andreas Wieland was working with Christian in my lab at that time. Andreas actually did something quite creative on his own. He also looked at B cells. He had enough enough samples remaining so he could do some B cell analysis. And I'll just summarize his work in one word by saying that there's also a very active B cell response ongoing to HPV. So these two papers were published back to back. This is the HPV-specific B cells in head and neck cancer, and this is the CD8 T cell paper that I summarized. So here are the findings from the CD8 T cell paper, that the stem, transitory, exhausted are there. They followed this different trajectory. And I did not show you the TCR chronotype data, but they are highly conserved. So the TCRs between stem, transitory, exhausted are completely shared because one is giving rise to the other. But the most important thing for us in following up is that the cellular machinery that could respond to PD-1 blockade actually exists in these individuals. So these treatment-naive HPV HNSCD patients do have a reasonable frequency of HPV and CD8 T cells. Not only that, but most importantly, they have the resource cells there which will respond after PD-1 blockade. The other important finding that came out of this is that E2 and E5 are major target antigens for the CD8 T cell response. Pretty much all of the effort currently on HPV has been with E6, E7. E6, E7 for vaccines, E6 and E7 for T cell therapy, everything E6, E7. This is a good target antigen because they are the oncogenes, but based on our limited data, I say limited because we didn't do a very large number of patients. Okay. E2, E5 actually are really quite significant targets. I think it would be good to include E2, E5 in these therapeutic approaches. Okay. And then I just want to end by one comment about PD-1 therapy treatment of PD-1 therapy of treatment-naive patients. So we are about to start, we are in collaboration with Roche. We will be starting a clinical trial of HPV-positive head and neck cancer patients who have not had any surgery or any treatment yet. Our prediction is that these patients might actually show a good response to PD-1 blockade. So this, and if the early clinical data, once we get it, is promising, this could be another treatment option for people. Instead of having the surgery, if the responses are promising, then this is something we're quite excited about this. And Roche is also quite excited in working with that. It may take us a couple of years, but we will be enrolling treatment-naive HPV positive head and neck cancer patients, and then treating them with PD-1 blockade prior to the surgery, and just following and see what happens, how these patients respond. Chuanbo, how am I doing for time? How much more time do I have?
You take as much as you want.
All right, I won't take too long.
It's okay.
All right,
It's okay.
I'll try to wrap this up in about 15
No,
minutes.
it's all right.
Okay, all right. So, so I want to kind of end on kind of a rational design for immunotherapy, because now that we have this understanding of these three states, and I want to emphasize that there could be more states found within these three states. But I think that everyone agrees that these are really very different cells. This is your quiescent resource cells sitting in niches. These are the early emerging effectors that will come from it, and probably this is the most functional of these cells. And then you have the more classically exhausted cells. So there are many questions here. What can one do for increasing number of stem-like cells? And then more importantly, can you do something for these cells? Currently, that's been probably the hardest target. But the question I want to address is, can we do something for improving the quality of the effector cells? That is, can we make them more like acute effectors? In other words, can the exhaustion program be changed? Because when you do PD-1 blockade, you get many more of these cells. And with PD-1 blockade at the target site, that helps and you get good viral reduction and you get tumor therapy. But transcriptionally and epigenetically, these three cell populations remain the same. That is, the transcriptional program and the epigenetic program of the transitory effector cells pre and post treatment is very similar. You have many more of them, but the program is the same. And the question we want to address is, can you change the quality of the effector CD8 T cells that come from the stem-like cells? So this is the story I'll kind of end on. And this paper was recently published, work done by Masao Hashimoto. And basically this shows that PD-1 combination therapy with IL-2 can actually modify the CD8 T cell exhaustion program and give you much more efficient or much qualitatively much better effector CD8 T cells. So the model we have used here, so everything we've used here is the LCMV mouse chronic infection model. And the first slide I'll show you is that this combination therapy of PD-1 plus IL-2 is very, very efficient. Of all the combination therapies we have tried in our mouse model, this is the best in terms of expansion of CD8 T cells and also reduction of viral load. And Erin West, who was a student in the lab almost like 10 years back, had published the original paper describing it. But this is data from Masao, and we have here 4 groups on this. This is looking at the LCMV-specific CD8 T cell response in the spleen. You see the same thing in other tissues, but I'm just showing you spleen. Here are the number of antigen-specific cells in untreated mice, PD-1 blockade, whether you use anti-PD-L1 or anti-PD-1, you see the same result. You get about a 5-fold to 8-fold increase in the number of cells. IL-2 by itself also causes an increase, but you'll see that the viral reduction only happens here. So, but if you do combination therapy, now you get almost a 10 to 50-fold. You can see this is untreated, this is anti-PD-L1, this is IL-2, and this is a log scale, so significant increase. And then this is the second tetramer, untreated, and the combination therapy, almost a 10 to 50-fold increase. And if you look at viral load, you get a reduction after PD-1 blockade, really not much reduction after IL-2, even though there is increase, because again, you don't have that blockade at the target site. Okay? But now if you look at the combination, really over 2 log drop. This is a very stringent model that we use. So getting a 2 log drop or a 1.5 log drop is really quite, is very impressive. So the question we then asked was, which cells are actually responding? Is this response coming from the stem-like cells, or is the response coming from the more differentiated cells when we do this PD-1 plus IL-2 combination therapy? Okay. And, oh, sorry, I'm a little bit ahead of myself. The next slide will show you that this viral control is mediated by CD8 T cells. So if you have untreated mouse, here's the viral titer in the spleen, here's the titer in the liver. If you do the combination therapy, you see this over a 1.5 log drop in the titer, in the spleen and the liver. But if at the time of the PD-1 blockade, you also deplete the CD8 T cells, so all viral reduction is gone. So what this shows is that we're expanding the CD8 T cells, and if you deplete them, you don't get any viral reduction, i.e. they are the main mediators of this viral control. So the key question was, when you're getting this huge expansion, after combination therapy, which cells are actually proliferating after PD-1 and IL-2 therapy. So the way this was done is to start with a chronically infected mouse, and you separate by using appropriate markers the stem-like population from the spleen, or the more differentiated population. Again, in this case, we use CXDR5 and TIM3. This is the stem-like population. This is the more differentiated. And then these cells were adoptively transferred into an infection-matched chronically infected mouse, congenitally different from the recipient. So donor cells have a different congenic marker, so we can track those cells. And then one group of these mice are given no treatment, the other one get the combination treatment. Now, the same experiment was done by Sagen in Sagen's original paper, showing that the PD-1, after PD-1 blockade, response comes completely from the stem-like cell. In this case, we are asking, in this experiment, when you have combination treatment, what happens? And the answer is, again, pretty much the response, the proliferative burst comes almost exclusively from the stem-like cells. So here is the data. So after the PD-1 blockade for 2 weeks, we are now looking at the expansion of the donor population that we transfer. So if we transfer the stem-like cells, okay, here is the very few that you find in the untreated mice. And after the IL-2 plus PD-1 blockade, you find almost this is a huge increase in the number, like almost 1,000-fold increase in the number of these antigen-specific cells. Of course, they have differentiated now, okay, but the proliferation all comes from, and if you transfer the terminally differentiated, you get no proliferation at all. And not only do you get this huge expansion of cells in the spleen, but you also get in the liver, you also get in the lung, and the bone marrow. The model here, not the model, but the data here is when you transfer these stem-like cells, they again intravenously, they go to the spleen, and you find them again in the T cell zones. Now you do the PD-1 blockade, these cells will beautifully expand and differentiate. And you can see that this differentiation, that this burst comes only when you transfer the stem-like and not the more thermally differentiated. In other words, to make the conclusion for this, which will become important later, is that the progeny after the PD-1 plus IL-2 combination therapy is emerging from that stem-like population. So then we ask the more important question, is this progeny, i.e. these cells that have expanded after combination therapy, are they qualitatively similar or different from PD-1 monotherapy? So we sorted the population at 2 weeks after PD-1 blockade, and then did RNA-seq to look at their gene expression program. And the results were quite striking. So here we are comparing the RNA-seq of tetramer-positive cells from untreated mice or after PD-1 blockade. Some minor differences, but very similar. As I told you, when you do PD-1 blockade alone, you get more effector likes, more of the transit population, but they are transcription very similar. But when you do the treatment combination therapy, you see huge difference. This is actually a very large scale. This is a scale from 1 to 50-fold changes. You can see very striking changes in this after combination therapy compared to untreated. We then did single-cell RNA-seq, which will show you really very clearly how the clusters are changing. So this was just bulk. This was RNA-seq of total tetramer-positive cells. The next slide will show you RNA-seq of tetramer-solid cell at the single-cell level, the single-cell RNA-seq. I'll take a few seconds to explain this. So in yellow are the untreated. So you find this is the stem-like population. So you have the stem-like. This is more transitory, and the more differentiated population, you find this over here. After PD-1 blockade, again, you have the same subset. So you see that these kind of sit on top of each other, the untreated and the PD-1 blockade. But when you do the combination therapy, you create a totally unique cluster. So there's a new cluster which accounts for almost 90% of these cells, is now a totally unique gene expression program. So what genes are different? That's shown on the next slide. So this is now looking at several inhibitory receptors, effector molecules, some other key things. So this is the unique cluster. This is the PD-1 plus IL-2 combination cluster. One striking thing is that TIM3 now is very low on these cells. If in the exhaustion program, TIM3 goes up on the transitory cells, stays up on the exhausted cells. So this is the stem-like cells, they're TIM3 negative, but the progeny is all TIM3 positive. But if you provide IL-2 signals plus PD-1 blockade, TIM3 is dramatically downregulated. PD-1 is still there on all of them, but TOX also gets slightly downregulated, and this NR4A2, which is again an exhaustion marker, is downregulated. So in other words, several inhibitory receptors, which you find in the transitory population and the more exhausted, are now downregulated in the combination therapy. But effector molecules are there. The granzyme B is there. Here, these are TIM3 was not there, but granzyme B is in, i.e., they are effector-like cells. KLF2 is very high in these cells. S1PR1, because they will come out into the blood and go to sites of infection, is very high. Very high here compared to even these the single blockade. A very interesting difference is IL-18 receptor. Again, IL-18 receptor is a very good marker of good effector-like cells. Good effector, seeded effector cells have IL-18 receptor. These have very high levels of IL-18 receptor. But now this is the part where it gets really interesting. So in the normal course of the program, which would be this, okay, and these are you, is that TCF1 is only on the stem-like cells and not on the more differentiated. But with the combination blockade, you even have a few granzyme B positive cells that express TCF1. And this is now looking more like the acute program. So the acute program, by acute program, I mean giving my, say, strain of HCME that will clear, looking at the day 8 effector-like cells, those, there is a, what we refer to as the memory precursor subset, has granzyme B and TCF1. So really, quite strikingly, this is now generating a very different type of cell. These cells also have LEF1, have more CD127. Interesting that they have some of these interferon-regulated genes. A very different beast has been created by providing this IL-2 signal. This is now looking at, that was transcriptional program, and our paper has many more details about all the genes which are different between the different, between untreated, the single therapy and the combination therapy. But also the ATAC-seq is strikingly different. This is looking at the ATAC-seq analysis of antigen-specific untreated versus anti-PD-L1 blockades, i.e. monotherapy. Very some changes, but very similar. But striking changes epigenetically in the untreated versus the combination therapy. Also, if you do this comparison of combination with mono, again, it will look as different as this. So what are some of the things that change? Again, the changes here are quite striking. For example, many of the inhibitory receptors are less accessible. For example, TOX is less accessible. And many of the more effector-like molecules like IL-18 receptor, others are much more open. So the ATAC-seq changes are consistent with the changes we see in the transcript level. And this is now an ATAC-seq PCA looking at naive cells that sit by themselves. These are all the different chronically infected cells, the different subsets of those, the stem-like, the transitory, and so on. And also after PD-1 monotherapy, they sit in this box. These are the acute infections. The day 8 effector cells, the terminal effectors, the memory precursors, and also the memory cells, central memory, effector memory. They have their own program. See how different the acute program is from the chronic program. And this combination therapy is giving us something in the middle, that they're moving more towards the, so striking change in the differentiation trajectory. So this is, I want to emphasize this. We are not changing, we are not rewiring a differentiated cell. So it's not like changing the transcriptional program of the exhausted cell. No, we're not doing that. What we're doing is we are changing the differentiation trajectory of the stem-like cells. The stem-like cells are going in this direction, but with the IL-2 plus PD-1 blockade, they're going in a better direction. So the effect is happening at the stem-like cell, not at the exhaustive cell. But it shows you how immunotherapy can be used to change that trajectory in the right direction. So basically, this summarizes it, that you have that these cells after combination therapy have more effector-like signature, decreased exhaustion signature. And then I'll end with this last part, which addresses a issue that's very relevant for some of the clinical trials that are going on. So we asked the question, is CD25 engagement important for the synergy that we have described between IL-2 and PD-1? The reason this is very clinically relevant is that there are many, many clinical trials going on right now, by the dozens actually, if not more, of cancer patients, where the IL-2 that's used is mutated, so it does not bind CD25. It still binds the beta chain and the gamma chain, but the binding to alpha chain has been done in such a way that it doesn't bind it. That goes like, and the reasons for this make some sense, actually make a lot of sense, because they're concerned about toxicity, and that's one of the reasons why they abolished binding to CD25. They're also concerned about activating Tregs. That's another reason. But we asked a very mechanistic question here, that in this model, where we are getting this beautiful synergy between PD-1 and IL-2, where not only are we getting very good viral reduction, but we are also generating CD8 T cells, which have a very different effector phenotype, i.e. much better effectors. Does this require CD25 engagement? So we did three things here. First, I'll show you a slide looking at the expression of CD25 in vivo after the combination blockade or combination therapy. And then I will show you data that what happens if you block CD25. That is, do the combination therapy and block CD25. And then I'll show you, lastly, what happens if you use a mutated IL-2. That is, an IL-2 that binds the gamma and the beta chain, but not the CD25, but not the alpha chain. All right. So first, I'll show you the upregulation of CD25. So this is, again, looking in a chronically infected mouse that's either untreated or given monotherapy or given combination therapy. So if you look at CD25 expression in untreated mice, actually very minimal expression of CD25 on any of these antigen-specific cells. This is all gated on tetramer positive cells, looking at CD25 and Ki67. If you do PD-1 blockade, you see that there is a significant increase in proliferation, 20% here, about 50% here, as expected, because they're proliferating, differentiating, but very minimal increase of 25. This is the combination therapy. Now pretty much look at the proliferation difference. Here it was about 50% of the cells. Now here you've got almost 90% of the cells are divided. And almost the majority of the dividing cells express high levels of CD25. So, and you also get upregulation to some extent, not so striking, of CD122 and CD132. So basically what the cells are now expressing is the high affinity IL-2 trimeric receptor. This is the real receptor that captures IL-2. It's not the beta gamma by itself, even though the signal is through beta gamma, and not the IL-2 by itself, not the alpha by itself, it's the trimeric receptor. If you go back and read all the classic papers done describing the receptor, the CD25 by itself has minimal binding to IL-2, very low affinity. Beta and gamma, slightly better affinity, but the trimeric receptor, which will only come up when you provide IL-2 plus CD25 signals, that is the real receptor that captures the IL-2. That's the orders of magnitude higher affinity. So not only do any cell with a trimeric receptor will not only capture IL-2, but also will keep it for longer. So it captures IL-2 and there's also longer durability of the signal. Okay, so the next key question was, what about what happens if you do the same combination therapy I've shown you that works well, but at that time block CD25? So here we got a very nice antibody that is very efficient in blocking CD25. And this is a antibody that blocks, doesn't deplete, so very effectively is being shown very nicely in the literature. So here is looking at CD8 T cells, antigen-specific CD8 T cells, untreated mice, nice expansion. Sorry, this is the level. PD-1 alone, monotherapy, you see nice expansion. If you do combination therapy, tenfold more. But if you now block CD25, all synergy gone. So, and if you look at the viral titer, the same thing. This is viral titer in the spleen in untreated mice after PD-1 blockade drop. Combination therapy much more. But if you block 25 signals, just like PD-1 alone, liver same way. Drop PD-1, bigger drop with the combination, but you block 25, just like PD-1 alone. And lung the same thing. So 25 engagement is critical for the synergistic expansion and also the viral load. And then the final experiment that I show you is using a mutated cytokine. That is the kind of cytokine which is currently very much in the clinic. There are some variations of it, but the main thing is don't bind to CD25 because that's bad. And so we got this mutated IL-2B from Roche, and you see that, again, it doesn't do anything to the antigen-specific responses. Untreated mice, PD-1 alone, combination with the wild-type IL-2, very nice synergy. With the mutated IL-2B, no synergy at all. And this is expansion of CD8 T cells. This is viral reduction in 3 different tissues. Again, the synergy is gone with IL-2B. That is, you don't see neither the expansion is there of antigen, nor is the viral reduction. So IL-2B is not, sorry, this one just basically shows that plays an essential role in this thing. But I want to point out to you that that IL-2B is biologically very active in vivo. That data is in our paper, and I'm not showing to you, is that it will expand total CD8 T cells. In fact, the overall expansion of total CD8 T cells with IL-2B is very similar to IL-2 wild type. We give a higher dose of IL-2B. We give 10 micrograms compared to 2 micrograms. Sorry, we give 20 micrograms compared to IL-2 alone, which is only 2 micrograms. Even with 10 times more, you get no expansion in cells, but total CD8 T cells expand beautifully. And the reason is very simple. Every T cell in the body has beta and gamma. So this is just picked up by all the cells which had a really minimal use to the tumor or to the chronic infection. So massive expansion of CD8 T cells, but minimal expansion of the antigen-specific is the classic sink effect. It's just being taken up by all the other cells. And I don't know how many of you follow the IL-2 clinical trials, but you may have heard of Nectar, which at one point had some promising data. It was pegylated IL-2 that BMS had actually invested very heavily in and bought the Nectar platform for $1 billion. And that was a complete bust. So the company is gone and the trial failed. It was pegylated IL-2, which was pegylation was done in such a way to block binding to CD25. So I think this is something for people to, in this area, to think about. I fully appreciate the concerns about potential concerns about toxicity and Tregs, but if you mutate IL-2 CD25 binding, the risk is it will just be diluted out. It will bind to every T cell in the body because all T cells are beta and gamma, and the ability of these cells to bind to antigen-specific cells, which would be to CD25, is lost. So another approach to this is targeting IL-2 to the right cell. And there was a companion paper with Masao's paper, and we worked with Roche, and this paper is back-to-back with ours, where they basically are using an IL-2B, but it's targeting, it's bound to anti-PD-1. So essentially, so in that case, the IL-2B will go straight onto will only bind to the cells which have PD-1 positive, and that is a reasonable alternative. So I think this whole area of cytokine IL-2 therapy, which is, I think, one of the most promising things, because the fundamental principle of T-cell biology and T-cell expansion and effector differentiation is that this is your effector cytokine. IL-7, IL-15, many others are homeostatic cytokines, very good for other things, but the classic effector cytokine is IL-2, and it works on cells that express CD25. So let me now finish with where I started, which is how do CD8 T cells adapt to a chronic infection? And I think they do this by having this very unique cell population that lives in a niche and acts as a resource. And now data has come out from many sources that this is a common theme for chronic infections when we first defined it, and also in cancer, as we and others have shown. And now a lot of papers are coming out showing that it's also true for autoimmunity. So I think that whenever you have chronic antigen stimulation, it can be in the context of a chronic infection, it can be in the context of cancer or autoimmunity. You need this resource cell, either for good things or for bad things in the case of autoimmunity. And if you ask the question in humans, where do you find tertiary lymphoid structures? You find them in autoimmunity, you find them in cancer, you find them in chronic infection, because that's where these cells will be residing in addition to residing in appropriate draining lymph nodes. Okay, let me end here by acknowledging many wonderful collaborators, but I want to just highlight a few people from the lab that played a major role. Sajan actually described these cells, when Hudson described the pathway more by identifying the transitory, and then Christiane and Andreas did the HPV work. Rebecca Obeng is the pathologist who's now on the faculty at Case Western. She did the very nice microscopy I showed you of the tertiary lymphoid vector. And Masao really an amazing effort for the IL-2 studies. Thank you very much.